All right, I'm Eli Roberson. I'm here from Washington University. I'm going to talk a little bit about a more basic question: what happens to human cells if they have low amounts of Nicastrin? I don't have any conflicts to disclose to you. Some of the things I'm going to talk about and be describing some ways in which we can perturb the expression of specific genes, how we can profile those. In this case, it's with transcriptome profiling. And that even if you're working at the level of single specific kinds of cells, it's still, the answer is usually it's complicated rather than simple. Now, when I was still a postdoc, somebody had approached my mentor about a project involving HS. I'm a human geneticist, not a clinician. I had never heard of it, so I was ready to write it off as somebody's pet project. And then I started looking, and the literature on the genetics of HS is actually really interesting, starting from the first studies that describe gamma secretase mutations, leading, of course, to a whole flood of studies afterwards where everyone is genotyped. And to me, when I started looking at this from the genetics perspective, the answer wasn't it's obviously got to be Notch. It's that the families that have the most solid diagnosis of having solved the mutation show a loss of function of Nicastrin. They're small indels that lead to nonsense-mediated decay. So I wanted to know what happens to cells if they don't have enough Nicastrin, not knowing what the effect should be. We didn't actually have any patient cells. So instead, what we decided to use was to go to some cell line studies and be able to knock down Nicastrin specifically. We decided to use two different cell lines, HEK001s that are human epidermal keratinocytes, and human embryonic kidney cells, HEK293s. They tend to grow well. As far as our method for knocking it down, we use short hairpin RNAs. It's a way we can introduce a plasma that produces a short hairpin that gets loaded into the RNA-induced silencing complex and basically just leads to the transcript being chewed up. So we haven't changed the genome, we just knocked down the level of expression. We had our core make us 5 different constructs that might work to knock down Nicastrin. We transduced all of them into these cell lines using modified lentivirus. And then whenever we actually tested for expression versus wild-type Nicastrin, the A6 and A7 looked like they didn't work at all in either cell line. All this, the level of 1 is just what you would expect from a cell that wasn't knocked down at all. If you look at A10, it looks like it kind of works in the 293 cells, but both the A08 and A9 constructs worked in both of the different cell lines. The A08 seemed to work better, so we decided to use that for our experiments going forward. Now, whenever you do this kind of transduction, even though it is highly efficient, you still get a mixed population. Some cells have your shRNA plasmid and some cells don't. We wanted to purify that out. So the plasmid with the shRNA also has a resistance construct to G418. So we took our transduced cells and we treated them with G418 for a week, so it's to kill all the cells that don't have a short hairpin construct, leaving only the cells that actually have the shRNA we're interested in. Now, not just having the A08 knockdown for Nicastrin, but also one against luciferase that's not in the human genome that should serve as a control just for the conditions. So after 1 week, you can see that the knockdown's even better. Now we're down to about 5% of normal level in the keratinocytes and about 20% of normal level in the embryonic kidney cells. So we had noticed while we were growing them that over time they just didn't look as healthy. If you ever had to do cell culture, sometimes you look at the cells and they're just a little weird. And we wanted to know if there was some of the weirdness was because of proliferation defects. We did a non-radioactive proliferation assay, and it turns out if you seed 3,000 cells per well onto a multi-well plate, you can grow them up over a week, and during exponential growth, there's no difference between the two at all. But as soon as you start to reach contact inhibition, the cells that have less Nicastrin begin to die faster. That's statistically significant for all points past where you start to see cell death. Now that was one interesting thing, but we also wanted to know just overall what's happening to these cells when they had less Nicastrin. So we had collected the RNA, we used Illumina beta rays to try to assay the expression of all the known genes in the human genome, comparing the A08 knockdown of Nicastrin to this background knockdown of luciferase, which should really not do anything. And if you just knock down Nicastrin, you get about 1,500 genes that show significantly different expression. We didn't know exactly what to expect. Notch might be one of the things that we could have expected to have come up at this point. And out of that 1,500, about half of them are a twofold change. I guess it's probably closer to a third of them, or at least a twofold change. So these aren't small effects with no other additional stimulus. Having a list of genes is great. Seeing these kind of volcano plots is pretty common in transcription papers, where on the x-axis you've got how big the effect of the change is. On the y-axis you've got the significance, so things that are way out and further up are highly significant in large effect sizes. But alone it doesn't tell you much. We can go in and do clustering too and look at the heat map. It shows mostly the advantage of using cell lines. It clearly separates all the cell cultures that have been knocked down with our control versus being knocked down with A08, and that there is clearly a set of genes that go up if you knock down Nicastrin and a set of genes that go up if you knock down Nicastrin. There's not a lot of variability. We almost never see this when we actually work with human tissues because obviously mixed tissues are a lot more variable than these cell lines are. Now we wanted to get something a little more useful from this. The main question was, what happens if you have less Nicastrin than you expect? And we can take this list of genes that we know are significantly differentially expressed, take the things that go up and use known pathways to say, do we see genes within a known function that accumulate more than you would expect by chance, meaning that a whole pathway's been disrupted. With no other changes to the cells at all, we actually get an enormous signal for type 1 interferon. The top signal actually is not Notch, and I might have even expected that it might be TNF-alpha or NF-kappa B because of the effectiveness of adalimumab, but it's actually an interferon response, at least within these keratinocyte cell lines. If you look at the genes that go down, there's actually a big increase in pathways associated with mitosis and the mitotic spindle, so we actually have some physical evidence from the assays showing that we have a growth defect backed up by the pathways that are disrupted whenever we knock things down. Now, I was a little confused that there was no signal at all from TNF in this case. I mean, we've done previous work in psoriasis looking at keratinocytes, and keratinocytes actually can do some local signaling, especially with things like TNF-alpha activating the NF-kappa B pathway. So we took the knockdown cells, and it's on the left side, and we decided to take them and transfect in an NF-kappa B responsive luciferase construct. So if the NF-kappa B pathway gets turned on, like TNF-alpha can do, you're going to see a higher signal. The interesting thing is that at baseline, there's actually a significant difference. If you knock down Nicastrin, you already have more NF-kappa B activity with nothing else going on. If you induce them, there's no difference. So there on the right of the left-hand plot, you see that everything is overlapping because they're not different at all. We wanted to know if this was a specific effect of this knockdown construct. Sometimes there's off-target effects. We wanted to see if something else was going on. So we used CRISPR-Cas9 mediated genome editing. We're able to make a HEC293 line that has a heterozygous indel in Nicastrin, which is not exactly the same indel that patients have, but it does obliterate half the function of the gene. Now at baseline, there's no difference on the right-hand plot of the expression of the NF-kappa B pathway if you have wild-type cells versus these heterozygous deleted cells. But if you induce with TNF-alpha, you see a significant increase in NF-kappa B signaling if you stimulate the cells with TNF-alpha. So this raises a couple of questions, one of which is if you have a proliferation defect with less Nicastrin, you really believe that having less Nicastrin is important in familial HS. High turnover cell populations could be affected by this, especially cells maybe in the skin or in the hair follicle. And if you're seeing something like the example we have of TNF-alpha showing increased activation of the NF-kappa B pathway only in the setting of being stimulated with TNF-alpha. Once you are exposed to something that might activate your TNF-alpha pathway, you're going to have longer lived and higher levels of inflammation than you would expect from a control. But some of the take-homes would just be, we do have methods. In this case, it was short hairpin RNAs and genome editing to be able to perturb single genes in a way that's really tractable in the lab. But the responses are cell type specific. If you look at between the keratinocytes, the embryonic kidney cells, there's 200 genes or so that have significant expression in opposite directions. So even in individual cells, this starts to get really complicated. And the cell lines are great, but they also are usually like hypotriploid. They have chaotic genomes. We really need to work more with induced pluripotent stem cells or directly with patient tissues. Things that we have going on now, we're looking more at using primary cells to look at the effects of loss of Nicastrin or other of these pathways, or stimulating with interferon to see what happens using single cell sequencing so that we could tractably use these cells. I have a postdoc in the lab that's developed a DNA sequencing assay that uses a targeted capture for all the components of gamma secretase with the components of the Notch pathway, Notch ligands, so that we can screen large populations of HS patients to identify anyone that not just has a deletion in Nicastrin, one of the other genes, but also regulatory variants that we might not have picked up otherwise. And we have an ongoing project with OSU Dermatology where we're getting patient punch biopsies and we're doing RNA-seq and microbiome profiling using long-read sequencing to try to figure out what's going on at an actual patient tissue level, which is going to be more complicated, but probably also more informative overall. And that's all I've got.